Dr. Shaheen Begum Madam, Associate Professor from the Institute of Pharmaceutical Technology, IPT, Sri Padmavati Mahila Viswavidyalaya, Tirupati, the one and only Women's University in Andhra Pradesh. Dr. Shaheen Begum Madam secured her bachelor's degree and master's degree from the University College of Pharmaceutical Sciences, Kakatiya University, Warangal. Madam worked as doctoral researcher under the esteemed guidance of our beloved guide, Professor Dr. Koganti Bharati Madam at IPT SPMVV, Tirupati, and got her PhD degree in 2017. Dr. Shaheen Madam has 18 years of teaching and professional experience for both the UG and PG students in the campus. By using a learning management system, which was designed by the Andhra Pradesh government system by LMS Portal Services, Madam monitored and delivered the specialization subject medicinal chemistry. Dr. Shaheen Begum Madam has co-authored 10 textbook chapters, 30 publications in peer-reviewed journals, and around 40 publications in peer-reviewed presentations in national and international conferences and workshops. Ma'am has supervised more than 50 UG and PG students in the campus, and Dr. Shaheen Madam has participated in 2 research grants as a coordinator. Dr. Shaheen Madam has a profound experience and expertise in the synthesis of new chemical molecules and entities by using simple laboratory techniques and also by using microwave-assisted irradiation techniques, and also has a strong profile and potential for the screening and evaluation of newly synthesized molecules for anti-inflammatory, anti-cancer, analgesic, and antipyretic activities in both in vivo, in vitro, and also in silico studies. And Madam has a profound experience in the research experience in various fields like in silico studies, QSAR analysis, both the QD and UDSAR analysis by using various software techniques. At present, Madam is thoroughly engaged actively in the design and development of novel antiseptic, anti-nociceptive, anti-cancer, and antibacterial agents using various in silico approaches. Madam is also effectively managing various polymorphism and associated technologies, and also investigating in drug-associated plasma protein binding interactions. I am very happy and thankful to our management and principal sir for giving me this opportunity of introducing my beloved teacher, Dr. Shaheen Begum Madam, who extended her helping hand in my research work, in my execution and the publication of my research work. Thank you in the campus. Thank you, Madam. Now I am welcoming Shaheen Madam for the presentation. Madam, kindly start the presentation.
Thank you so much, Dr. Anita, for a nice introduction. So, I'm also thankful to your organizations at REDDY College of Pharmaceutical Sciences for giving me this opportunity to present some of the work. Shall I share my PPT?
Yes, ma'am. Sure.
Is it visible?
Yes, ma'am. Please full screen.
Yeah, yes. I'm converting it. Is it clear? Yeah. Yes, I have made it to PowerPoint presentation. Now, is it clear, sir?
Full screen.
Screen, is it in PowerPoint? Is it in presentation mode? Still, I have.
Mode only, madam.
Yes, sir. I have shared only presentation mode only, but I think it is not visible. Yeah. Yes. Now is it clear?
Madam, PPT notes.
Sir, I kept in PowerPoint presentation mode only. Alt, 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 Control, Tab. Okay. 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 Yes, yes. Alt, Control, Tab. Yeah, yes, yes, yes. Yeah. Okay. Now, is it there? I have shared it. Now I need to press, or after sharing, we need to click the windows, no? Mm. Okay. Okay. Hello, sir. I will mail my PPT. So it is not coming in my system. Let us see. I am mailing to Dr. Anita's mail ID. Yes, sir. I have sent to mail ID. Please share from that desktop.
మేడం మెయిల్ చేయండి మేడం కంటెంట్ ని నేను షేర్ చేస్తా స్క్రీన్ మీద.
చేశానండి.
ఓకే మేడం.
Yes. What happened now?
Hello.
Hello.
Madam.
Yes.
Madam, I have shared the screen, ma'am, your presentation. Is it visible, ma'am?
No, not yet. It is not yet visible. Yes, now it is visible.
Okay, ma'am, you can continue.
Yeah, sorry for the inconvenience, but I was unable to upload it from my desktop. So we will start our presentation. Today I will try to discuss on RNA, various forms of RNA and ADAR enzymes. That is adenosine deaminase acting on RNA and their role in human diseases. Coming to the contents, I'll discuss about transcription and translation, and then post-translational changes that are taking place in RNA, various different forms of RNA, RNA metabolism, degradation, and also RNA modifications or RNA editing. Next, I will focus on ADAR enzymes and that will lead to A to I, that is adenine to adenosine to inosine editing, and then the therapeutic importance of these processes or ADAR enzyme. And I'll try to focus on what is the research scope for the budding faculty from this topic. Yes. Next slide. We are all aware of transcription and translation, which are very fundamental for the growth of a cell or any cell, that is whether it is a plant or human, whatever it is. These two transcription and translational processes are fundamental for the cell. So what is transcription? That is conversion or formation of mRNA from DNA is called as transcription, whereas coding of proteins, different types of proteins from mRNA is called as translation. Slide please. So we all are very much aware of these processes, which are because these are fundamental. We know that mRNA is formed from DNA and polypeptides, and then various kinds of proteins are produced from mRNA by coding process. That is called as translation. But it is not so simple. Now the research is very tremendously has taken place in nucleic acid research, and scientists have found out what are the various in detail processes that are involved in the post-translation transcriptional processing of mRNA. Next slide. So here you can see the difference between bacterium and eukaryotic cell. Here in the first diagram, you can see that DNA is giving our mRNA. The from the double helix structure of DNA, you can see that there is a formation of RNA transcript. So the first mRNA that is formed from DNA is called as RNA transcript. Okay, it is called as RNA transcript. In eukaryotes, this transcript is not mature enough so that it can go to cytosol and perform in for protein synthesis. It is not at all ready. So it needs to undergo a lot of processing. So that is called as post-transcription changes in the mRNA. That is very much required for eukaryotic cell. Whereas in bacterium, as soon as RNA transcript is formed from DNA, it directly enters into cytosol and involved in protein synthesis. So this is the basic fundamental difference between the mRNA that is available in bacteria and mRNA that is formed in eukaryotic cell. It means that mRNA transcript needs to be processed little further so that it can perform protein synthesis. Let us see in detail what is the pre-mRNA processing. Next slide, please. So here the post-translational processing, it involves a lot of modifications, chemical modifications. First, we will see the basic structure of DNA. DNA is having promoter region and also DNA coding sequence. Okay, so this is the fundamental part of DNA that is involved in mRNA synthesis. Here, synthesis of mRNA is carried out with the help of RNA polymerase II enzyme. And what happens to this pre-mRNA? The 5' region of mRNA, 5' region of mRNA undergoes capping, and then once 20 to 30 nucleotides are assembled to form a mRNA, immediately capping takes place here at the 5' end. And you can see that exon and introns are available in the pre-mRNA structure. 5' UTR and 3' UTR. These blue colored regions are untranslated regions. Okay, these regions are called as untranslated region, and the middle part is called as translated region. So it is translated from the DNA, and here some part of the RNA, that is pre-mRNA, it is having UTRs at the 5' end and the 3' end. Okay, here finally once 20 to 30 nucleotides are assembled, finally polyadenylation takes place at the 3' end. Okay, you have to remember these two processes. 5' end undergoes capping and 3' undergoes polyadenylation, that is a formation of tail. So this pre-mRNA again goes converts to mRNA. So here it is given 5' cap is capping takes place with the help of 7 methylated guanine. So here the 7th position of guanine is involved in the formation of a bridge with the 5' end, and a triphosphate link is formed. So this 5' bridge is essential for the normal working of a functional RNA. Okay, so here UTR again, it is an untranslated region. At the end it is having polyadenylated tail. So it means pre-mRNA undergoes polyadenylation, 5' capping, and also splicing. Splicing means all the introns are removed, which are non-coding sequences, and all the exons are ligated, which are coding sequences. And finally 3' processing, and also you can see A to I editing takes place in the RNA processing. So we are today discussing about A to I editing. So it is a part of RNA processing. You should remember that A to I editing is a part of RNA processing. Coming to the various forms of RNA, we are aware of mRNA, rRNA, tRNA, but if you see the classification, now the research has given so many RNAs. There are nearly 20 types of RNAs are available in our body, which are recognized and whose functions are very clearly defined. Except 2 or 3 RNAs, all the other RNAs, their functions are clearly defined by the researchers. So hopefully we will see all these types of RNA. The protein coding RNA is called as messenger RNA. That is mRNA, that is synthesized in nucleus by DNA. Okay, so coming to the other non-protein coding RNAs, these are referred to as ncRNAs. Here you can see there are 17 types of RNAs I have given. I'll just read out some of them. Small nuclear RNAs, snRNAs, snORNAs, siRNA, saRNA, miRNA, and shRNA, like the pY associated RNA ribozymes, exosomal RNA, and eRNA, that is enhancer RNA, and also scRNA, that is a small Kajal. So these many RNAs are available in our body, and let us see how they function and what are their therapeutic role. Next slide. So if you see the content wise, the mRNA is constituted around 5%, 5 to 10%, and majority of is rRNA. And the other RNAs, they exist in very, the percentage of their constitution is very less. They are available in fractions. The percentage of their occurrence is very little. The mRNAs, it is important in protein coding. As we have discussed, mRNA is essential for the protein coding process. And ncRNA is involved in protein synthesis. And snRNA, it plays very important role as a component of snRNP, that is RNA binding proteins. And like this, these all RNAs, they exist, and some of them are situated in nucleus, and some of them, depending upon their function, they are available in cytoplasm. Here you can see that the nucleus is having a rRNA, snORNA, like that. RNA precursors are there, and also spliceosomes, which are made up of snRNA. These two are available in nucleus, and you can see that there are several RNAs are there: long non-coding RNAs, microRNAs, small interfering RNAs, and pY associated RNAs that are referred to as pRNA. These are all important in RNA interference pathway or gene silencing pathway. They are available in cytoplasm, and the RNA is required for the translation, that is coding into protein process. We all know them: mRNA, tRNA, and rRNA. They are available in cytoplasm. And exosomal RNA, they also exist. Exosomes are vehicles. Exosomes and endosomes are available in our cell. They are the small vesicles. And exosomal RNA, it means some RNA parts like mRNA and miRNA, they also exist in the form of exosomal RNA. So this is the localization of RNA. Let us see the life of RNA. Once RNA is released from the nucleus, what happens, and what exactly take place even in the nucleus, we will see. And then we will discuss about RNA degradation and then RNA editing. Next slide. So coming to RNA metabolism, as I said, capping, splicing, and polyadenylation. These three processes are very, very important for a newly formed RNA transcript, so that it can become a mature mRNA and it enters into cytoplasm for the coating process. Here you can see that figure A. Here the eRNA is also there, that is enhancer RNA. It controls the process of genetic expression, and we know that how mRNA forms, but it is also evident from the research that nucleus also give rise to several other non-coding RNAs. So here long non-coding RNA, red colored RNA, which is having a shape like a stand like a shape, and here circular RNA, which are covalently bound RNA. These two also, they are synthesized in the nucleus. So what happens to these RNAs, why they are important and why they are formed means, these are associated with to control the genetic expression. It means to control the RNA levels, to control the other RNA levels. They are formed in the nucleus. They are associated with DROSHA and DGCRP, and they combine especially long non-coding RNA, LNC RNA. They combine with DROSHA and DGCR8, and they give rise to mRNA. So these pre-mRNA, once they are formed in the nucleus, they enter into cytoplasm. So here you can see that not only mRNA, but LNC RNA, circular RNA, and pre-miRNA. These are the 4 types of RNAs which are synthesized in nucleus, in cell nucleus. They are also released in cytoplasm. We know the role of mRNA. Once mRNA is released, the coding part, it means whatever the translated region is there, that will translate into protein at the endoplasmic reticulum, and the other mRNA which is not translated, that will be converted to P body or stress granule. Okay, this P body is very, very important mRNA decay machinery. So some of the mRNA is stored in the cytoplasm as P body or Kajal bodies also, and stress granules. And what about the other circular DNA and LNC RNA or miRNA? They are very cleverly, they are introduced in the cytoplasm and they negotiate gene expressing. Once pre-miRNA is entered into cytoplasm, it will associate with argonaute proteins, PI proteins. They are very important in negative feedback, that is, they stop the genetic expression. So they combine with AGO and stop the process of mRNA and coding process. So here protein synthesis and mRNA synthesis is controlled by this RISC. So this is all taken place in the cell RNA metabolism. So let us have a closer look on what is the RISC, that is the RNA-induced silencing complex. This is actually a multi-protein complex. As you can see, it is made up of dicer, it is made up of TRBP, it is having argonautes also. TRBP is actually 3 double-stranded RNA domain, binding domain, and dicer is classified as RNase 3 endonuclease enzyme. So combinedly, these 3, when they are associated with double-stranded RNA, they have negative expression in the gene expression. They stop the process of generation of RNA. So that is why it is called as RNA interference mechanism, or it is also called as gene silencing mechanism. Here not only dsRNA but also some single-stranded RNA also, they participate in the RNA interference pathway. So after metabolism, we'll see what is how degradation takes place for RNA. Once the RNA is there, once that concentration of RNA is high, DNA undergoes degradation. Here CCR4 not and XRN1, these are the 2 essential factors which helps in degradation process of RNA. Carbon catabolite repression -1 Tartales A multiprotein complex, it is called as CCR4 not, and also XRN1, it is a protein that is exoribonuclease 1. They all decap. We have seen 5' capping and adenylation process that is important for the functional RNA. Here you can see that the capping is reversed, that is decapping is taking place, and also adenylation is reversed or de-adenylation takes place. All the adenine molecules are removed from the mRNA tail, 3' tail, and now this mRNA become non-functional mRNA. So it is called as a de-adenylation dependent pathway. Then the another pathway by which RNA undergoes degradation is endonuclease dependent pathway. Here endonuclease cleaves the adenylation part and cleaves it so that it is not functional. So these are the two essential pathways for the degradation of RNA. Next. So we have discussed about what are the various forms of RNAs and what exactly takes place, how they are synthesized in a nucleus and how they are metabolized, what are the various forms which are available in the nucleus and how they enter into cytoplasm, and how some part of the mRNA is involved in gene expression at the cytoplasmic level. Another important process that takes place in RNA is RNA editing. It is also called as RNA modification. Actually, it is evolutionary conserved property of RNA. It takes place by nature. There are in almost plants, animals, and eukaryotes. Everywhere, wherever there is RNA undergoes some level of editing. Some part of RNA undergoes editing. So it has a lot of physiological significance as well as they are important for diseases also. So how they undergo RNA, how RNA undergoes addition means it takes place only at the level of nucleotides. Okay, RNA is made up of, we know that it is made up of nucleobases, ribose, sugar and phosphate backbone. But evolutionary conserved properties, they allow the modification at the nucleobase level only. So here there are 4 mechanisms: insertion, deletion and deaminases. These 3, they change structure of RNA. Let us see the basic structure of DNA and RNA, how they differ. We are all very much aware, but I'll try to just focus on again. DNA is double helix structure and RNA is single helix structure. So it is having single strand, whereas DNA is double stranded. So one of the reason which might be DNA is closed means the information is packed in DNA, but whereas that is not necessary for RNA. Here the nucleobases are very free. It will go and interact with so many other, and that information is coded somewhere. The base pairs that are involved in DNA are 2 pyrimidine bases that are thymine, cytosine, and purine bases are adenine and guanine. Whereas in RNA, the thymine is replaced with uracil, and the another pyrimidine base is cytosine. The purine bases are adenine and guanine, the same which are available in the DNA. So here, if you have a closer look on the structure of thymine and cytosine, they both differ only in the methyl group. Thymine is having a ketone group at the 4th position, and cytosine is having amino group, and thymine is having one methyl group at the 5th position. And here uracil and thymine, they differ only in having CH3 group. Okay, uracil is not having any CH3 group at 5th position, whereas thymine is having CH3 group at 5th position. That is the only structural difference between thymine and uracil. Yes. Next slide, please. So as I said, RNA editing occurs in majority of the cells. So we can see modified nucleobases in the cell cytoplasm as well as in the nucleus. Here the adenine and guanine, they will be modified to hypoxanthine, xanthine, and also 7-methylguanosine. And pyrimidines, here you can see 5,6-dihydrouracil is formed, 5-methylcytosine and 5-hydroxymethylcytosine. These are all 6. Here I have mentioned 6 nucleobases which are structurally modified, and these are present in our human body. In human body, even in other mammals, in eukaryotic cells, all these nucleobases are available, and because of RNA editing. Okay, so this is the structure of RNA. It is made up of nucleobase, ribose sugar, and phosphate link, phosphate backbone. The carbon which is associated or which is linked with nitrogen base is actually numbered as 1. So the ribose of second, the position, RNA is having ribose sugar. Here RNA is having 2 hydroxyl group at 2' position, whereas in DNA you cannot find this OH group. That's why it is called as deoxyribose nuclease, nucleic acid. Sorry. Okay, so this is the fundamental difference between RNA and DNA. The 3' end and 5' end, these are OH, and here the third carbon is called as 3' end, and the fifth carbon is called as in the ribose is called as 5' carbon position. Okay, next slide. Now there are so many experimental and computational approaches are available to obtain the 3D structure of RNA. There are plenty of biophysical approaches like X-ray crystallography, NMR, and cryo-EM. And assays is actually neutron assay, and more details of the RNA structure, like envelope structure. Its phase distance distribution between RNA is also calculated with EPR, XAS, and SM-FRED technologies. Not only this, but with the help of these biophysical, also by using computational modeling techniques, also we can predict the structure of RNA. We will again go back to RNA editing mechanism because we are focusing on A to I editing. The first RNA modification was first described in 1968, where RNA methylation was observed in HeLa cells. In an RNA molecule, it is with the delivery of in the RNA, and because of the deaminases. There are enzymes which remove amino group from the nucleobases. So the main editing mechanisms are insertion or ligases and effect of deaminases. Next. Yes, here you can see that uridine or uracil, the nucleobase, it may undergo first cleaved, it will be removed, and the both again they are ligated with the help of RNA ligase. Similarly, once cleaved, these uridine bases maybe they can be added to the already existing mRNA to give a modified base. So this is the process by which uridine addition or deletion takes place, and because of which there will be totally change in the structure of RNA. Next slide, please. So what happens if at all RNA editing takes place, and why it is essential? We know that other than 4, that is adenine, uracil, cytosine, and guanine, nearly 150 different kinds of chemically altered residues are available in these cells. And that is very important. Maybe they are very, very important for the RNA structure and to maintain the stability and dynamics of the RNA structure and RNA splicing mechanism, polyadenylation mechanism, and also transport of RNA and localization of RNA, and also connections of the RNA editing may also take place in rRNA structure, So there will be translational efficiency will be RNA editing takes place in. Okay, so fundamentally RNA editing takes place in different kinds of RNA, but it will be for the use of our to attain or to control the situations whenever there is a viral attack, whenever there is a potential pathogens, whenever they enter into the system, or whenever any organ undergoes any disease. These changes may take place in different structures of RNAs. So the various RNAs and how RNA editing takes place. Now ssRNAs that are single stranded RNAs and double, but few RNA they may exist as double stranded. They're encoded as dsRNA. So dsRNA is having two complementary strands, and it can be again by RNA dependent RNA polymerase enzyme, and then you compare with single stranded RNA. And dsRNA is most of the times it is there in the viruses, or dsRNA is generated whenever the virus invades host cell, and during the replication process also dsRNA is generated. So it plays very important role in the innate immunity of the system. Single-stranded RNA, there are single-stranded RNA is depending upon its polarity, it is again classified into positive sense RNA and negative sense RNA. Positive sense RNA, it is having mRNA which is functional and it can encode its own proteins. But negative RNA, it needs to undergo sense RNA. It should be converted to plus RNA, and then only that RNA will become functional to give protein synthesis or to synthesize proteins. So other than this dsRNA, it is there in several bacteria, rotavirus, reovirus. These RNAs, they are having dsRNA. We all know that SARS-CoV virus, it is also involved in the reaction. So water, the H2O is involved, and one of the oxygen of this water molecule, it enters and it attacks on the cytosine ring, and whenever amino group is removed, this O group attacks on the carbon, and a ketone, that is uracil, is formed from cytosine. So deamination of cytosine will lead to formation of uracil. Next, the important is A to I editing, that is conversion of adenine to inosine. Previous slide. Here adenosine molecule is now, here the same thing, amino group is removed, and at that place CO group is introduced in the adenine molecule. So this reaction takes place via adenosine deaminase acting on RNA. That is the our discussion is taking place on this enzyme ADAR. So this enzyme is crucial for the conversion of adenosine to inosine. It is, next slide. So this A to I editing takes place via adenosine deaminase acting on RNA enzyme. This enzyme binds only to double-stranded RNA. It is very important point to remember. Our body is having ADAR1 and other isoforms also, which can deaminase the adenosine to inosine, but it acts only on double-stranded RNA. Whenever it comes in contact with single-stranded RNA, there will be no action. It will act only on double-stranded RNA. Okay, so wherever there are double-stranded RNAs in our normal cells, ADAR also goes and attacks on that molecule and edits the mRNA transcripts. So majority of the times A2 editing sites are introns, that is the important area of RNA, that is pre-mRNA. Untranslated regions that I have showed in the first slides, UTR, 5' UTR and 3' UTRs. Non-coding RNAs are also sites of A to I editing. And majority of the times, sine and line, these are the repetitive elements that are present in RNA molecule. Those are also important sites for the ADAR enzyme. So ADAR enzymes are available in different isoforms in humans. They are ADAR1, ADAR2 and ADAR3. Next. So what happens if at all adenosine is converted to inosine? So we know that adenosine or adenine, it combines with uridine with a double bond. We know that ATCG is there in DNA base pairs, and AUCG base pairs are available in RNA. So adenosine is having a lot of preference to uridine in normal circumstances. But what happens if it is converted into inosine? So this is the structure of inosine. Here it is having a CO group. So it resembles to guanosine or guanine. Here you can see that guanine is having a CO group here at this position, similar to inosine. So there is a confusion between whether the nucleobase is inosine or whether it is guanosine. And similar to guanosine, the formed inosine base pairs with cytidine. So here actually it should interact with uridine, but after conversion it will pair with cytidine. So there is a mismatch. So difference between how adenosine interacts and how inosine interacts with the base pairs. And there is a lot of impact with this ADAR enzyme. If it is not working properly, or if it is not expressed, if it is upregulated or if it is downregulated, the levels of adenosine and inosine, they will change. And wherever in whatever the physiology or pathophysiology, this adenosine is important and ionics are important, this ADAR enzyme plays a lot of importance, and that's why it is therapeutically very, very significant. So what happens, for example, this is for just for the more clarity, because I told you that ADAR acts only on the double-stranded portion of RNA. I thought it will be clear for you that in the RNA structure, they will be recoding, and definitely it will have a lot of impact on the RNA structure and stability. Yes. Next, please. So there are, I told you there are so many isoforms for the ADAR enzyme, ADAR1, P150 and P100, ADAR2, ADAR3. These are all available in our body, but the therapeutic significance is there for ADAR P100, P150 enzyme, because it is expressed whenever there is a viral attack or any host, any pathogens, they enter into our body. So you have to remember this one isoform, ADAR1 P100. 100 is actually constitute, but P150 is the one which is expressed whenever there is a host invades our body. So it is having zinc DNA binding domain and DS binding domain, and also deaminase binding domain.
It is audible, ma'am?
Shall I continue?
Yes, ma'am.
Yes. Yes, madam.
Next slide, please. Yes. So these are the interpretation of the dsRNA which are coming from the outside of the cell or inside of the cell. ADAR enzyme is also reported to inhibit the replication of hepatitis C and also HIV. By adenine is there, the coding will have a different, and their channels will be largely controlled by these enzymes. And it is also important for the multi-organ growth. So the dsRNA contains adenosine. Because it is edited, it will have inosine in its at its site. It will go and bind with RLRs, MDA5 and RAG1, and it will inhibit the process of interferon release. Okay, what happens during a viral attack means during the viral infection, or whenever there is ADAR1 is inactive, or whenever it is inhibited, the dsRNA goes and binds with the MD5 and other cellular signaling receptors, and it will induce the process of interferon response. So ADAR inhibition is required whenever there is a host enters into the, whenever a pathogen enters into the host. Okay. Next. So ADAR is actually, its role is complex in various cancers. Sometimes it is promotes cancer, and sometimes in some cases it also suppresses cancer. So in some of the cancers like prostate cancer, ADAR1 levels are upregulated. Likewise, in bladder cancer, ADAR levels are downregulated. So in this condition, whenever upregulation is there, we can inhibit the ADAR enzyme, and whenever downregulation is required, we can upregulate by using their analogs. Yes, next please. Next please. So these are the this I have taken from a recent review where oncogenic long non-coding RNAs are given. So some of them are oncogenic, some of them are having suppression activity on tumors, and H119 is actually having both the oncogenic and tumor suppressor role. So the role of ADA1 and these non-coding RNAs are very crucial in different types of cancers. So this is just to show the importance of A2I editing. Here you can see that AMPA receptor, whenever it is unedited, it is permeable to calcium ions. But once edited, once adenosine is converted to inosine, the edited RNA, it will give a receptor, it will produce a receptor, it will change the receptor so as it is impermeable to calcium. So these roles are very, very important for the normal functioning of ion channels in the brain. So ADAR2 enzyme is involved in the neurotransmitter controls. 5-HT receptor, here you can also see that unedited is having permeability, but edited is having no permeability. Similarly, the functions of channels also, they change with the editing of adenosine to inosine. So on the whole, I would like to tell that ADAR1 and ADAR2, both of them, they are the upcoming and very important targets for controlling immunity, for controlling cancers, and also for controlling some of the degenerative diseases. So after thorough investigations, few of the ADAR inhibitors were synthesized and their activity on ADAR enzyme was measured. Some of the ADAR inhibitors are non-selective, but they have similar structure when compared to adenosine. That is 8-aza-adenosine. It is having one extra nitrogen in the imidazole group, and 8-chloroadenosine, it is having chlorine at the 8th position of adenine ring. So these are the two, because they are having lot of structure similarity with adenosine, they were tried as ADAR inhibitors, but they are non-selective. Next. So another analog which is based on the structure of adenine, it is called as EHNA, that is erythronine 2-hydroxy-3-nonyl adenine hydrochloride. It is also tested in several studies as ADAR inhibitor. Its selectivity is also less towards ADAR1 enzyme. So these two, the above mentioned two slides, that is non-selective inhibitors and EHNA, These two are only investigated as ADAR enzyme inhibitors. Other than this, several investigations in silico based on the molecular docking, virtual screening, and dynamics were also proposed. And for example, recently we can see a study here where they have used the ADAR enzyme structure that is available as PDB. The PDB ID is 2GXB. This is actually Z alpha binding domain of ADAR1. So using this structure, we can virtually screen, and by the in silico investigations, we can investigate or we can design some of the ADAR inhibitors. Some bisphosphonate class of compounds were investigated from the computational studies, and they showed greater ADAR inhibitory activity when they were researched. Next. So other than these, translational, depending upon the translational state of ADAR, that is enzyme, a few of the ADAR inhibitors were designed. Here if you see the intermediate structure, it is having a tetrahedral intermediate. So based upon the mechanism also, several ADAR inhibitors were designed. Next slide. So here 8-azane bularin is the compound which was which showed ADAR inhibitory activity by mimicking the transition state of the ADAR RNA editing reaction. So these are the fundamentally very less research has been carried out on this topic, and there is lot can be done, and there is a lot of scope for the investigations. We can do lot of work on ADAR enzyme. Next slide, please. So this is just I have mentioned what is the scope. So because there is a PDB crystal structure is available for ADAR1 enzyme, we can do docking studies and dynamics, and also we can if the compound is showing any potential docking score, and if it is potentially active in virtual work, then we can go for definitely synthesis of that. That is the one I can suggest. And we can also design and synthesize transition state analogs based on, because a transition state crystal structure is available, and we know the mechanism. Because of that also we can design the novel molecules, and we can also try natural compounds as ADAR enzyme inhibitors. So thank you so much. This is all I have to discuss with you.
Thank you, ma'am. Any queries, please? If anybody have any queries.
Yes, actually it is not our normal medicinal chemistry, but it is a potential area who want to work in the computational work. For example, if you want to plan any in silico work, definitely you can design, because it is a novel area. Very few scientists they are working on that area. Till now there are no reports. Only very few compounds have that ability to inhibit that ADAR enzymes, and especially it is useful in cancer treatment.
Shall we wait for 5 minutes, ma'am? Anybody will ask any questions?
Yeah, sure.
Any queries, please, from anybody? Yeah. Let's check on that. can ask, sir.
See, online it is very difficult to interact, but I tried my level best to make understand the topic.
Yes, ma'am. Actually, it is somewhat a tough topic, ma'am,
Yes, yes,
for understanding. Okay.
yes, yes.
Thank you for your valuable and knowledgeable session, ma'am. First and foremost, I extend my sincere gratitude to our esteemed guest speaker, whose presence has added immense value and prestige to this program. Your insightful remarks have inspired us to strive for excellence and have provided us with invaluable guidance for the journey ahead, ma'am. On behalf of Sir C.R. Reddy College of Pharmaceutical Sciences, I extend my heartfelt appreciation to all the participants who made this event a resounding success. My sincere thanks to our management and principal, sir, for providing me this opportunity. Thank you, ma'am. Thank you for sparing your valuable time.
And I am also thankful for giving me the opportunity on my behalf and on the behalf of IPT, SPMVV. I would like to thank all the authorities of Sir C.R. Reddy College of Pharmacy. I'm very thankful to the organizers, and I'm also thankful for the participants who were very calm enough and who participated in the class. Thank you so much.
Thank you, ma'am. Thank you, everyone. Have a nice day.